starting off, just general features. When you think about what the kidneys do just in general, their job is to take about 20 to 25% of the cardiac output and run it through a series of little tiny tubules. You have over 1 million of them in the kidneys. And these little tubules are called nephrons, and their job is to basically balance the dissolved electrolytes that are contained within the blood, and then also deal with the removal of metabolites and waste products. In doing so, we also will be able to balance the water content of the blood, so we can regulate blood pressure and blood volume. We can get rid of excess hydrogen ions, so you can regulate blood pH. And last but not least, the kidney, like many of our organs, has a hormone function, and the kidney releases some really important hormones that are responsible for regulation of blood cell production. So we have erythropoietin, which will stimulate bone marrow hemopoietic cells, which are stem cells for blood, to develop into red blood cells. Renin actually is an enzyme that gets released that begins that process of converting angiotensinogen into angiotensin 1, that then gets converted by ACE in the lungs, which you talked a little bit about, into angiotensin 2. So renin actually begins that process, and the kidneys will regulate the volume of blood flow going into the kidney to determine whether or not the pressure, the amount of blood going into the kidney is appropriate, and whether or not we need to save more water. And then we also have another hormone called calcitrol, which is involved in regulating calcium levels. So first and foremost, when we look at whether or not the kidney is doing its job, one of the easy ways to assess kidney function is to look at urine. Urine essentially is the product of the kidneys filtering and reabsorption. So if the kidneys are doing a good job, they're going to make an appropriate amount of urine. Urine is, obviously it's a waste product, but it's a very informative one, because if you think about it, urine was circulating as part of blood maybe 30 minutes to an hour or so prior. Depends on the last time you voided your bladder, but it's basically a subset of what is in blood. Essentially, it is a blood filtrate minus important water that gets retained, important electrolytes that get retained, and then obviously the nutrients that get balanced by the liver, we want to make sure that those get retained. So what's in urine is anything that's excess. So excess water, excess electrolytes, organic waste, and organic nutrient waste. So what I want to go through is a little bit of how we do that regulation. We'll spend more time in lecture dealing with this. But this figure is showing you one of the little tiny microscopic tubules that takes a very small amount of blood and starts separating out what's important from what is waste. And we call those little tiny microscopic tubules nephrons. The very first part of that process is going to be a filtration, which is simply a separation by size. That occurs here in the nephron. So this structure is also called the renal corpuscle, or renal body. This is an enlarged view of it. And it actually has a little tuft of blood vessels called the glomerulus, sitting inside an epithelial cap here called Bowman's capsule. And we'll see that blood will flow into it, containing in it valuable, important nutrients that have been balanced by the liver, but also excess electrolytes, water, and waste product. And so the first thing that we're going to do is we're going to separate what's important from what is excess through this filtration process. And anything that's large, by default, is going to be important. So those are things like blood cells and large plasma proteins. Those are pretty big. So they will not be able to be filtered through the tiny little gaps within these little blood vessel cells. And so if you were a blood cell coming in, actually maybe I should use a different color because it's hard to see red, but let's say you were a blood cell coming in, and I'm going to make it blue because that's easier to see. So here's a blood cell coming in. that blood cell is too big, so it'll kind of bounce around and navigate its way around through this tuft and then back out again. Same thing would be true for proteins. So if you're a protein, like one involved in clotting, you don't want to lose your clotting proteins. And so let's say this is a clotting protein. They're also pretty big. Those are going to go in and they're going to bounce around in here and eventually they'll go out. But if you were a salt, like maybe sodium, those are going to be tiny. So sodium, for example, will go in and then it gets slipped through and across the endothelial cell, the blood vessel, and then they get caught in this space. So sodium will be able to exit. Sodium is an electrolyte. Same thing will be true for potassium and magnesium and calcium. All those are small electrolytes. They'll all get filtered across the tiny holes. If you are a nutrient, let's say your glucose, let me make that cyan. Glucose is also pretty small. It has to be absorbed across our metabolically active cells. And so here we're going to see that glucose also slips across. Now, if glucose slips across, that's actually not necessarily a good thing because remember the liver would have done its job in balancing the appropriate amount of glucose. So glucose should be at 90 milligrams per deciliter coming in. And we want to, that doesn't really look like glucose, but we want to make sure that that glucose stays in the blood in that same amount. And so the problem for glucose is that it's going to get filtered. It's not going to stay in the blood vessel. Unlike the proteins in the blood cells, they're too big. They'll actually stay within the blood vessel and go back through the renal vein back into general circulation. But if your glucose or any of the amino acids or any of the important electrolytes, you're going to be too small to get retained. And so you're going to get filtered into this Bowman's capsule or renal capsule. And then the problem is that you'll then be on your way through this renal tubule. It's kind of a convoluted or twisted pathway. I'm going to change the color just simply to green so I can see it a little better. Let me find green here. There you go. So let's say you were a glucose molecule, it'd be going down through this convoluted pathway and back up again. If you don't get reabsorbed, you end up going all the way around and then out. And if you go out, then you're part of what we call a urine if you don't get reabsorbed. So anything we want to retain in the blood, if it's small and gets filtered, has to be put back into the surrounding blood vessels. So we're going to find that the nephron does an amazing job of reabsorption. And so that's what we're going to see here. All these blood vessels that surround this tubule are going to be picking up anything that gets reabsorbed that we don't want to pee out or be part of urine. Anything that doesn't get retained and picked back up and put into surrounding blood, if it travels through this convoluted pathway and then out, it then just simply gets excreted as part of urine. So that's kind of the quick and dirty. Basically, you're going to do a blood filtration first and then a filtrate reabsorption. So we're going to filter out all the tiny things, retaining all of the big constituents of blood, which are always going to be important. Anything big is going to be important. So blood cells and formed plasma constituents. So those will be your proteins and the proteins that are carrying hormones. Those are all going to be retained. And anything that's small, those will be your electrolytes and your small nutrients, as well as waste products. Those are going to get filtered. And so if you want to retain them, you don't want to lose them as part of your urine, then you have to reabsorb. And so that's this tremendous job here is reabsorption. All right, so let me move to the next slide. Macroscopically, the urinary system is super straightforward. That's one of the easiest anatomical systems to learn. So structurally, we have two kidneys. Each does essentially the same job. So collectively, they'll be filtering about every 40 minutes your entire blood volume, which is crazy. With every heartbeat, 20-25% of your cardiac output is going to be moving through your kidneys. Urine production rate is going to essentially result in urine getting produced where we filter about 1 liter of blood every minute. We're going to have about 180 liters of filtrate produced in a day. Most of that filtrate gets reabsorbed, so typically the volume of urine produced in a day is about 1 to 2 liters. Urine capacity can be up to 1 liter, but most of us will void or urinate before the bladder fills that much. So in most cases, we're going to void when we get to about 1/2 liter or so. And that's dependent on the sensitivity of our stretch receptors. All right, so we have 2 kidneys. These will be filtering blood, so they're blood filters. They, alongside the liver, are basically the 2 major organs that regulate the constituents of our blood that circulate to every cell, at least within a millimeter. So the liver does the job of regulating the nutrient composition of blood, and the kidney's job is to regulate the electrolyte and water composition of blood. So when the kidneys are doing their job, we're going to see that the result or the product of that filtration and reabsorption is going to be whatever's left over is urine. That's produced all the time. We produce urine all day long. The volume really depends mainly on your hydration state. The more hydrated you are, the more water you'll excrete to help maintain an adequate blood pressure. But that urine would then collect in these foot-long muscular tubes. So these are called the ureters. They do peristalsis just like the digestive system does peristalsis. So we're going to see intercircular and adlongitudinal muscles that massage urine down toward the bladder. The bladder is also made of muscle. It's actually made of 3 layers of muscle. You can have the ability to get more holistic contraction and voiding with those 3 layers of muscle. So the bladder is where we're going to store urine that's produced all day long until appropriate time of voiding. And then we have a single exit from the body. Both males and females have a single exit, which is called the urethra. In males, we'll also couple urethra with the reproductive system. All right, so 2 kidneys, 2 ureters, a single bladder, and a single urethra. So that's the macroscopic major structures for the urinary system. All right, so again, kidneys are your blood filtering, reabsorbing structures. We'll look more at the details of the histology in lecture. I'll show you some histological slides that allow you to view the different regions of the kidney. But again, their job is to regulate the electrolyte and water composition of the blood. So they make that homeostatic regulation of electrolytes and water. And the product of this process is going to be urine. So urine is anything that's in excess. Excess, we mean like water, excess electrolytes, and waste products. Ureters are muscular tubes. They're about 1 foot in length and travel the length of the body cavity from the kidneys all the way down into the pelvis. Their function is purely transport, so they aren't doing anything to the urine other than just simply transporting it to the inferior located bladder. You'll notice that the kidney on the right side sits a little bit lower, and the kidney on the left is a little bit higher, and that's simply due to the positioning of the liver on the right side. All right, so the bladder is a muscular chamber. Again, it's going to be for temporary storage till we can make sure that we have an appropriate time to void it. And then we have urethra. The urethra is the exit point from the bladder in both males and females. There are some gender differences. So I mentioned in males, the urethra functions both for urination as well as reproduction. So when an individual is ejaculating, there's actually an inner urethral sphincter that will close off the bladder so that the bladder will not be voiding urine at the same time it's ejaculating. The male urethra tends to be about 8 to 12 inches long. In females, the urethra is purely for urinary product. It exits the female and it's much shorter. So in females, it's about 1.5 inches to 2 inches. That's one of the reasons that females tend to get urinary tract infections more frequently, because the outside world is only about 1.5 inches to 2 inches away. So it's a much shorter distance for microbes to travel than in the male, which is about 8 to 12 inches away. All right. So here's a quick overview of the urinary system.
The renal system consists of the kidneys, the connecting arteries and veins, and the urinary tract. The kidneys are paired, bean-shaped organs lying outside the peritoneal cavity in the back of the upper abdomen, one on each side of the vertebral column. The kidneys function as the body's main excretory organs, eliminating the body's metabolic waste products by filtering the blood. Substances that are unneeded or are present in excess are filtered out of the blood and form into urine, moving via the ureter into the bladder before being expelled through the urethra. The kidneys also selectively reabsorb those substances that are needed to maintain the normal composition of the blood. By adjusting blood composition, the kidneys are able to maintain blood volume and pressure, ensure the proper balance of sodium, chloride, potassium, calcium, hydrogen, phosphate, and pH, and eliminate products of metabolism such as urea, uric acid, and creatinine. The medial border of the kidney is indented by a deep fissure called the hilus, where the blood vessels, nerves, and ureters connect to the kidney. The kidney is composed of up to 18 lobes. Each lobe is composed of nephrons. Nephrons are the functional units of the kidney, and each kidney contains more than 1 million nephrons. Each nephron consists of a glomerulus and a system of tubules. The glomerulus is a unique, high-pressure mass of capillaries that filters the blood. The glomerulus is encased in a thin, double-walled capsule called Bowman's capsule. The space inside the capsule and surrounding the glomerulus is called Bowman's space. Plasma-like fluid is filtered from the capillary blood into Bowman's space through the glomerular filtration membrane. The glomerular filtration membrane consists of 3 layers of capillary wall: the endothelium, the basement membrane, and the epithelium. This membrane allows some particles from the blood to pass through, but not all. The fluid that is filtered from the capillary blood into the Bowman's space is called filtrate and forms the primary urine. The filtrate then diffuses across Bowman's space and into the tubule system of the nephron. In the tubules, some substances are added to the filtrate as part of the urine formation, and some substances are reabsorbed out of the filtrate and back into the blood. The nephron tubule is divided into 4 segments. The filtrate passes through each of these segments before reaching the ureter. A highly coiled segment called the proximal convoluted tubule, which drains Bowman's capsule and where almost complete absorption of nutritionally important substances takes place. A thin looped structure called the loop of Henle, which reabsorbs water and ions from the urine and plays a role in controlling the concentration of urine. A distal coiled portion called the distal convoluted tubule, which regulates potassium, sodium, and pH, and where further dilution of the urine takes place. And the collecting tubule, which joins with several tubules to collect the filtrate and where final sodium regulation takes place. Each kidney is supplied with blood by a single renal artery that arises on its respective side of the aorta, before dividing into 5 segmental arteries that enter the hilus. Within the kidney, each segmental artery branches into several lobular arteries. The lobular arteries further subdivide to form interlobular arteries, which branch off into afferent arterioles. Blood flows into the glomeruli through the afferent arterioles. Blood flows out of the glomerulus through the efferent arteriole. The afferent and efferent arterioles regulate glomerular capillary pressure by selectively dilating or constricting. The kidney's venous blood, now filtered, flows from the glomerulus via the efferent arterioles into the peritubular capillary network, a low-pressure, reabsorptive system surrounding all portions of the tubules. This arrangement permits rapid movement of solutes and water between the fluid in the tubular lumen and the blood in the capillaries. The peritubular capillaries rejoin to form the venous channels by which blood leaves the kidneys and empties into the inferior vena cava. Urine formation involves the filtration of the blood by the glomerulus to form an ultrafiltrate of urine, the tubular reabsorption of electrolytes and nutrients needed to maintain the constancy of the internal environment, and the secretion of waste materials. Filtration occurs as blood flows into the glomerulus from its afferent arteriole, and plasma moves through the glomerular capillaries into Bowman's space. From Bowman's space, the glomerular filtrate moves into the tubular segments of the nephron. Here, through tubular reabsorption, Electrolytes and nutrients move from the filtrate back into the bloodstream. Here also, through tubular secretion, substances move from the peritubular capillaries into the urine filtrate. The filtrate concentrates in the collecting tubules, then finds its way to the renal pelvis, where it is directed to the ureter, the bladder, and the urethra for elimination. The kidneys perform an excretory function by filtering the blood and then selectively reabsorbing those materials that are needed to maintain a stable internal environment. The nephron is the functional unit of the kidney and is composed of a glomerulus, which filters the blood, and a tubular component, where necessary substances are reabsorbed into the bloodstream and unneeded materials are secreted into the tubular filtrate for elimination in urine.
Okay, so when we look at the kidney's location, the kidney, in fact, the entire urinary system is located in a position that's referred to as retroperitoneal, which I'll talk about in just a minute. Also looking at the kidney, it basically sits between the serous membrane that lines the body cavity and the abdominal musculature in the back. So when you think about the lumbar muscles, um, we have the extra-abdominal oblique, remember, coming off of these lower ribs, 5 through 12. We have some muscles that we didn't learn. So, for example, the lumboreus quadratus sits back in here. So when we look, we're going to see that the kidney is actually leaning against the body wall, the posterior musculature of the body wall, and it's actually technically outside the body cavity. We're going to find that the right kidney sits a little bit lower than does the left, and that's because the liver is over on the right side. So this is looking posteriorly at an individual. So you're looking from the back end. These dotted lines represent the iliac crests. So we can see that the kidneys are almost going to be sitting right on top of your iliac crests. This space here is actually going to be filled with a really nice cushion of adipose. So they're kind of sitting right on top of the iliac crest in a nice little pillow of adipose, leaning against the posterior abdominal wall, and then covered with a sheet. It's almost like, laying in bed with a sheet that kind of covers the kidneys on both sides. And not just the kidneys, but also the ureters, as well as the bladder, are all going to be technically outside the body cavity because they're going to be covered with the parietal peritoneum. All right, so if we look at their location, this has a little bit of the musculature intact. So we have some of our erector spinae muscles. This is the quadratus lumborum. We have our external abdominal obliques right here. Here they've kind of cut away some of that muscle so you can see the bottom edge of the kidney here. So if you're trying to approach the kidneys, the easiest way to actually approach the kidney is not from the front, but rather from the back. If you're approaching the kidney from the front and you're trying to, access the kidney, you have to go through all of the abdominal viscera before you finally get to the back of the body wall where the kidneys are really going to be adhered to. So it's like you'd be sort of delving within about a foot of, abdominal cavity organs before you finally hit the kidney. But if you go posteriorly, right between the lowest rib and the top of the iliac crest, once you go through this little sort of layer of musculature, you're going to be right on top of that kidney. So that's really important. They sit about the 11th or 12th rib on either side of the vertebral column, literally attached to the dorsal abdominal wall. All right, so I mentioned that they're what's considered retroperitoneal. Retro means behind, and they're talking about the peritoneum. In fact, all organs in the body start in a retroperitoneal position in the embryo and the fetus. And during fetal development, they're going to navigate forward. And as they move forward, they're going to pull that parietal peritoneum with them. And then we refer to it as the visceral peritoneum. And the stretch of the parietal peritoneum that gets pulled off the dorsal abdominal wall, that's going to be called the mesentery. The kidneys don't do that. The kidneys stay in their location. They don't move from where they're originally formed. And so they stay literally attached to the dorsal body wall with the parietal peritoneum running over them. So almost like attaching to the wall and then wallpapering them against the wall. So I always use this analogy when I was little, my sister and brother and I used to, you okay? You hurt your finger? No. Okay, I'm gonna go upstairs. Hold on. Okay. I was little, my sister and I shared a room, and my brother would always come into the room, and so we'd always get annoyed. So we used to always yell at my brother, "Mom, Mark's in our room." So then he would stand at our doorway and he'd stick his arm in the room. And so the question is, if you stick your arm in the room, are you technically in the room? It's kind of like it is for the kidney. When you look at the kidney, technically you're going to see the bulge of them. So if you move the abdominal viscera over, you're going to see these mounds that project into the body cavity, but technically they're not in it because they're going to be lined with this parietal peritoneum. So this is a drawing of that. So they've done a transverse or horizontal section through the abdomen. This is the rectus abdominis muscle here. These are the obliques. So there's your external abdominal oblique, internal abdominal oblique, and transverse abdominis. And we can see in red is what we refer to as the parietal peritoneum. So it's going to come across the abdominal wall. So that's that serous membrane. As we approach the dorsal abdominal wall, we're going to come off and around our organs. And so here we can see the kidney is sitting in this paravertebral recess, which we have on either side. Sometimes refer to this area as the paravertebral paravertebral gutter. That was the other term, paravertebral gutter on either side of vertebral column. So you have this sort of big indentation surrounding the kidney. This is all going to be adipose. So you have this perirenal or perinephric fat or adipose that sits all around it. And they refer to that as the adipose capsule. It functions to kind of cushion, protect, and hold the kidney against the dorsal body wall, along with this wallpaper that's going to go right over them. So that's true for not just the kidney, but also the ureter and the bladder. So the entire urinary system is considered retroperitoneal, technically outside the body cavity. All right. So when we look at the kidneys, they have a lot of connective tissue, loose connective tissue over them. And so when we look at the parietal peritoneum as it travels over the surface of those kidneys, we actually refer to that parietal peritoneum as renal fascia. So that's that fibrous connective tissue. It's going to be fairly easy if we were to dissect it to kind of break it with just tips of scissors or forceps. But it helps to secure and hold the kidney in place. This is a really nice drawing where they're highlighting the kidneys. You can see, here's the iliac crest on either side. You can kind of see the kidneys sit on top of the iliac crest on either side. This big muscle here, that's the psoas major muscle. And this is one of the large vessels that's returning blood back to the heart. So this is the vena cava. And on this one, they're not highlighting it in red, but this is the aorta right here. So that's the blood vessel that's bringing oxygen-rich blood down toward the kidneys. Now the kidneys are inside of this pad of adipose along either side of vertebral column. So we have the perivertebral gutters that are going to be filled with adipose, which we refer to as perinephric fat that makes what we call the adipose capsule. And then we're going to cover the kidneys with our parietal peritoneum, which we refer to as our renal fascia. All right, so this is a nice view of the perinephritic or perirenal fat, which we collectively refer to as the adipose capsule. If you were doing a dissection, you can literally pop the kidneys out and it leaves a depression where they were sitting in this adipose capsule. This is a really great view here from the side. So you can kind of see it's almost like you put a pillow against the backboard of your bed. If you've ever done that and watched TV, so you're sitting in bed with this nice pillow against the backboard. That's kind of what's happening here. We have this nice big pillow of adipose leaning against the back wall of the body cavity, sitting atop the iliac crests. So it kind of situated exactly on the posterior body wall. All right, so the adipose is this thick surrounding structural fat that helps to support and protect and cushion the kidneys, and then it's going to be covered with the renal fascia, which is just really the parietal peritoneum. All right, and then the last thing is the renal capsule. The renal capsule is super thick. It's very difficult to break it. In fact, you'd have to use a scalpel, and even with that, it's really hard to cut through. So it's a really thick, tight shrink wrapping of dense, irregular collagen fibers that almost look like shrink wrapping around the kidney. They help to support and maintain and protect the kidneys, maintain actually the shape and protect the kidneys. And so here in this drawing, they've kind of cut with a scalpel the renal capsule and kind of used a forceps to pull it off. If you were to touch here with the end of like a tweezer or forceps, you might leave a little bit of an indent. But if you were to touch here, you can actually poke all the way through. So this is really soft tissue underneath. This is where we're going to have all those little tiny microscopic tubules and blood vessels. So this is quite soft tissue relative. All right, so when we look at the superficial anatomy of the kidney, it is concave medially, and it's going to be convex laterally. And so when we look at this concavity here, very similar to the lung, we use the same terminology. So we call this the renal hilus or hilum. So depending, hilus is a little bit older term. Hilum is a newer term that we're using. So we'd say renal hilum or renal hilus. Plural would be hyla, so you'd have two of them. In this concavity, this is where the major tubing is going to be going in and out. So we have a renal artery going in, carrying blood to be processed. That blood will then branch into sequentially smaller and smaller vessels as it goes all the way out to the periphery of the kidney. And so at the periphery of the kidney is actually going to be the entry point into our blood filtering tubules that will then filter the blood as it travels toward the center and then back. And then when that blood is fully processed, the processed blood will then leave through a series of blood vessels that eventually merge into the renal vein that then exits. We also have one more tubule, which is going to be the ureter here. And this is then going to be tasked with carrying urine that's produced by the kidney during that filtration reabsorption process down toward the bladder. All right, so the renal hilus or hilum is the entrance and or exit, depending on the direction of fluid flow, of the major renal blood vessels. That's the renal artery carrying blood in and the renal vein carrying blood out, and the ureter, which is going to be carrying urine itself. The renal vein is actually anterior. The renal artery is posterior. So the renal artery is going to be behind the renal vein. And then kind of sandwiched in between them is the ureter. So you can see the ureter on both sides. All right. What we then would do was we would do a frontal section on our kidneys so we could see inside. The human kidney is very consistent to what you would see if you sectioned a pig kidney, which is why we would have done that this semester, and why I posted a video of a pig kidney dissection. But hopefully I'll get into class this week and I'll be able to videotape some of our human kidneys. But when you do a sectional anatomy, there's three regions that we identify. We have this outermost region in the periphery, which tends to be a little bit more granular in its appearance. Kind of looks like it has little nubbins in it. Tends to be a little bit lighter in appearance. And so this outside edge we call the renal cortex. This is actually where blood is eventually going to be directed. So blood comes in through the renal hilum, and it's going to branch and branch and branch, and its goal is to go all the way out to this outside periphery. And then you're going to enter into your renal tubules. The renal tubules are going to be organized into these pyramid-shaped lobules here. And so they call these the renal pyramids. Depending on where you look, the numbers are usually between 8 and 12 or 10 and upwards of 18 of these different pyramids. The numbering doesn't really make much difference because they all do essentially the same thing. And depending on an individual, the size of the person will determine the size of the kidney and typically how many lobules you have or pyramids you have. So the pyramids are sitting inside this sort of middle region called the renal medulla. They're separated by connective tissue, which helps to route blood vessels through. Those are called the renal columns. And then in the very center of the kidney here, at the access point for all of this tubing, is a space here called the renal sinus. And that space allows for the entry of these blood vessels and the exit of the ureter. The ureter's job is to catch all of the urine being produced by these different renal pyramids. And so at the tips of all these pyramids, you would imagine urine seeping from the tip and has to get caught in the collecting structure of the kidney. And they're going to sit in this renal space or renal sinus. And so we're going to talk about these collecting structures in just a minute. So we have three regions, the cortex, which is out here. This is where we're actually going to begin the process of blood filtration. We have the renal medulla, which consists of the pyramids, which are these kind of cone-shaped lobules, and the columns, which are going to be between them directing blood. So the medulla or middle is going to be pyramid and column. And then the last is the renal sinus, and the renal sinus here is the third region, is going to house the major tubing that's going to be coming in and out. And then kind of filling the spaces is going to be adipose, because we don't want these things wiggling around in there. We're going to pack them with adipose. That's why you see all this adipose or interior renal fat. All right, so cortex, right, outside periphery. Medulla is going to be that middle portion. So this is the medulla in the middle. And then we have the renal sinus, which is this interior hollowing, which allows for the tubing to come in. All right, so just a little brief on this. Like I mentioned, the renal cortex is this outside edge. This is where we're going to see blood is going to come in through the renal hilum or hilus. That blood is going to start branching out and going to move through the columns within the medulla all the way out to the cortex, where we're going to begin the process of filtration at the cortex. So again, it's going to look kind of granular. This is the site of blood filtration. So all those little renal corpuscles that we saw in the video in that one image I showed you, they're going to give this region of the kidney kind of that granular dotted appearance to it. The renal medulla has usually between 8 and 12 cone-shaped pyramids, but you'll see as few as 6 in some people, as much as 18 in others, so quite variable. Here we can see this is a human kidney, and they're showing you here this outside cortex, which typically is a little bit lighter. And then we have the pyramids, which tend to be darker just because, remember, when you're moving through those tubules, you're going to have a lot of blood vessels surrounding them to try and make sure that we can get reabsorption taking place for anything that the nephron would have filtered, but we want to make sure it doesn't become urine. So they tend to be a little bit darker in color because of the amount of blood vessels sitting there. And they tend to have more of a striped appearance or a linear appearance because of all the tubules. They're going to be arranged side by side, projecting toward the tip of the renal pyramid, which we call the papilla. All right, so the appearance is going to be very linear, and this is where we're going to have all of those little nephrons lined up side by side, trying to reabsorb any of the nutrients and electrolytes that got filtered that the body still wants to retain. All right, so when we look at a renal papilla, this is just simply the tip of the renal pyramid. So we look, here's a renal pyramid sitting in the medulla, and the tip of it they call the renal papilla. So remember, we've used that term papilla or papillae many times. Papillae is plural, papilla is singular. We just mean the cone-like projection. This is where essentially the product of the nephrons will be seeping from the collective work of all of the individual nephrons within that particular pyramid. So all of the nephrons are going to work together to make urine, and that urine is going to emerge from each of these sort of cone-like masses of those little urine-producing tubules or nephrons. All right, so I have here papilla is the tip of the pyramid, and these are going to release the urine. The columns are going to sit between the renal pyramids, and they're really just connective tissue helping to support blood vessels as they navigate all the way out to the cortex. So I have here renal columns are sitting between the pyramids. It's really just connective tissue that's going to function to help support the blood vessels as they travel to the cortex. Okay. All right, and here you can see them using that term hilum instead of hilus. So depending on where you view, you may see hilus or hilum. All right, so here they use the term hilus. The renal sinus is the last region, and so notice here, I was actually surprised when I saw this. When you cut a kidney open, you look inside, this center area is a big space that accommodates essentially the urine catching structure here. The ureter is going to expand out to this big funnel that's going to project toward each of the individual pyramids to catch the urine. So I have here, it's kind of like this big funnel that sits inside this interior space called the renal sinus. Okay? And then to make sure that these don't wobble around and move away from their connection, we're going to pack them with adipose. This will all be filled with adipose. All right, so this is a really nice anatomical drawing. So they're showing you the blood vessels way out here at the periphery. So blood is going to enter through the renal hilum. It's going to branch and branch and branch. It's going to go through the columns in the medulla and out to the cortex. In the cortex is where we're going to enter into the renal tubule structure that will then extend down and then back up through these renal pyramids. So this is where we're going to have all that reabsorption. Anything that doesn't get reabsorbed becomes urine, and then it's going to seep through the renal papilla and get caught by this urine collecting structure. All right, so again, we have cortex, medulla, and the sinus. And the sinus is going to contain the urine collecting structure, which I'm going to name in just a second, and the renal artery and vein. All right, so here's the urine collecting structures, which is really cool. So here's the ureter. Again, the ureter is going to be about 1 foot in length, and it is made up of smooth muscle to do peristalsis. As it approaches the kidney, it's going to expand into this funnel-like projection. So this section here, that's the expanse of the ureter, is called the renal pelvis. Pelvis means basin, so it's aptly named. So this is the pelvis, and then the pelvis is going to expand outward toward the individual pyramids. Now these pyramids are arranged three-dimensionally. So if you're taking your hands, put your palms together and interlace your fingers so that your fingers are still straight, but they are moving between each other. That's kind of like the orientation of these pyramids. So they kind of sit juxtaposed to each other. So each of these pyramids has to have a collecting structure associated with its papillae. And so that's what we see here. This is going to be what we refer to as the calyx structure. A calyx is a twist. And so as we approach each of the pyramids, we're going to have what's called a minor calyx associated with the papilla. So it's going to be like this little cup here around each of the papilla. That's referred to as the minor calyx. And then the minor calyxes are going to collect urine and drain it into what we call a major calyx. So this is a major calyx here, and then into the renal pelvis. So a minor calyx is associated with each papilla from a single pyramid. They're going to collect urine from each pyramid and then direct it to the renal major calyx. Okay, so this is looking here. Here's a pyramid, and here's a pyramid, here's a pyramid. And so these regions of collecting structures around them are called the minor calyx. The major calyx is going to be the union of the minor calyxes. This is a really great three-dimensional image. You can see how they're sitting kind of juxtaposed to one another, so they kind of fan out within the kidney, so they're not all in the same linear plane. So we have, in this case, 1, 2, 3 pyramids that would be draining into these minor calyxes. The minor calyx would drain into the major calyx that then drains into the pelvis. And that's true across all of the renal pyramids. All right, so this is just a nice view of that again. So these are minor calyxes draining into a major calyx that then drains into the pelvis. So again, here's 2 more minor calyxes. You can see a little recess around the renal papillae, and the minor calyxes drain into a major calyx, which is right here, that drains into the renal pelvis. And what we're just really doing is collecting all that urine produced. And then the renal pelvis is then going to become the ureter as it exits the kidney's renal hilum. All right, blood supply. When we look at blood supply, we're going to have a major renal artery carrying blood that needs to be processed into the kidney. They approximate that about 20-25% of our cardiac output is going to travel into the kidney. Remember, that's about the same volume that's traveling into the liver. So if you have 20-25% of your cardiac output going to your liver and your kidney, that means that about half of your blood volume is going into these 2 major homeostatic organs, one regulating your nutrient load, one regulating your water and electrolyte load. And so that's really important to recognize. With the 2 kidneys functioning, collectively they're able to process your entire blood volume every 40 minutes. So if you had maybe asparagus as part of your Easter dinner or dinner, then you might be able to smell the breakdown product of the asparagus, which is quite odoriferous in your urine, even within 30 minutes of eating. So it's pretty quick how fast that travels through. They estimate that we're producing about filtering about 1 liter of blood every minute. That's a tremendous amount of filtration and balancing that takes place. All right, so just naming wise, the arteries are carrying blood into the kidney, the veins are carrying processed blood out of the kidneys, and the nice thing is that they have the same naming. So if you have an artery, you're going to find it has a counterpart vein that has the same name. So we have the renal artery and renal vein. The renal artery is going to travel in and it's going to branch into about 5 what we call segmentals. The segmental vessels are then going to branch into what we call the interlobars. So the interlobars, there we go, are then going to branch between the pyramids. So remember, these are called pyramids or lobes. And if you're going between them, that's referred to as the interlobars. So we have renal artery going into segmental, going into the interlobars. The interlobars are going to be sitting in the columns between the pyramids or the lobes. And then as we get to the cortex, we then need to go over the surface of the pyramid. So these are going to be the arcuates. And then we're going to go into the cortex. The cortex vessels are referred to as the cortical radiate arteries, or the older term, which is called interlobular. That's what we have here. Or interlobular. So we have interlobar between the pyramids, and then we have interlobular, which are now called cortical radiate. I really like cortical radiate better, one, because then I don't mix up interlobar and interlobular, which sound very similar, but you can see how these vessels are radiating out into the cortex, which is why we call them cortical radiate arteries. The veins are going to be named exactly the same thing. So once blood has been processed, it travels in the reverse direction. So we'd go down the cortical radiate veins to the arcuate veins, to the interlobar veins, to the segmental veins, and then out the renal vein. All right, so you want to make sure that you feel comfortable identifying those vessels as well as just looking at blood flow. So again, if you're at the renal artery, that's carrying blood that's to be processed. It goes into the renal hilum or hilus. It branches into about 5 segmental. This branching is going to occur in the sinus. They will then approach the pyramids in the medulla. As you get to the pyramids, the only way you're going to get between them to the cortex is to go through the columns. So we call those the interlobars. When you get through the columns between the pyramids, then you have to go around the surface of the pyramid. So those are called the arcuates because they arc over. And then once we get over the surface of the pyramid, we have the cortical radiate or the interlobular. And those are going to supply each of the pyramids with blood. All right, so again, the renal veins are just going to collect processed blood and run it in reverse. Okay? Nope, not yet, sweetie. No, no. All right, so, and then the blood gets processed and goes back to the heart. All right, so this is just a nice inset showing you blood with waste products arrives to the renal artery. That's going to travel in. It gets processed. Remember, the nephron is this little tiny blood filtering and reabsorbing structure. Anything that gets filtered and not reabsorbed becomes the waste product we call urine. That urine will then get collected and deposited into the series of collecting structures from the minor calyx to the major calyx to the pelvis and then the ureter. So here waste products drain toward the bladder through the ureter. And all of the processed or clean blood will then exit the kidneys through the reverse transport. So you're going to have the radiate cortical to the arcuate, to the interlobars, to the segmentals, to the renal vein, and then back to the heart. So now you have this wonderful clean blood, like we have here, clean blood leaving from this renal vein, and they'll dump right into the vessel that's going straight back to the heart. It's called the vena cava. Okay, just a tiny bit on the ureter, and we're almost done. The ureter is a smooth muscular tube. It propels urine from the kidneys all the way down the dorsal body wall, so it is a retroperitoneal. It's made up of 2 layers of muscle, so we have the inner circular and outer longitudinal layers of muscle, so it does effective peristalsis. And then it's going to enter the bladder posteriorly. Okay, so it's going to come into the back side of the bladder toward the base of it or the bottom of it. So we're actually going to fill the bladder from the bottom up rather than from the top down. You see in the back of the bladder here are the 2 openings for the ureters. We call these the 2 ureteral openings. It's really important to recognize those because the 2 ureteral openings form the corners of what is a triangle called a trigone here. Inside the bladder, we're going to notice it's actually really rough, and just like the stomach, which is going to store food that we eat until we can move it into the small intestines, urine is going to be stored here in the bladder, so the bladder has to stretch just like the stomach does. So we're actually going to see rugae in the interior of the bladder, except for this smooth funnel-like area called the trigone, which is going to be determined by the 2 ureteral openings and then the exit from the bladder and the entrance into the urethra. So that's this smooth area called the trigone. All right, so the 2 openings into the bladder are called ureteral openings. So we have a right and left ureteral opening. All right, so again, it's about 1 foot in length. It is retroperitoneal, so it would have parietal peritoneum covering it. It's made up of smooth muscle and transitional epithelium. So we have this ability to stretch without tearing the inside layer of the ureters. They can actually carry quite a significant volume, especially when you're really hydrated. All right, urinary bladder, again, is going to function to store urine. When we look at the bladder itself, it's the same in males and females. So female bladders are not bigger nor smaller than our males. The difference in how long you can hold your urine is really more a factor of the stretch receptors and your sensitivity to those. And when we look at the positioning of the male versus the female, we take in consideration the reproductive structures. So the bladder, just like the ureter and the kidney, are also retroperitoneal. So you'll see here in this drawing, this is the parietal peritoneum coming off the body wall and going right over the surface of the bladder. I mention that because I know when I did my first dissection, I had only studied anatomy from textbooks and models, and they make the bladder look like an insanely obvious structure. But in a cadaver, the bladder is going to be devoid or void of urine, so there's not going to be anything in it, and it's flattened against the body wall. So if you're looking for it, you're not actually going to see it because it doesn't bulge into the body cavity at all. So it just looks like a smooth, flat floor here. And when I first was looking, I was actually really embarrassed because I'm like, I can't find bladder. And then when you cut actually right in front of the bladder here, the parietal peritoneum, and look in, then you can actually see the bladder itself, and you can dissect it. But unless you actually cut through the parietal peritoneum, you're going to look like just a flat floor in the bottom of the pelvis. So with that said, in males, we look at the bladder. The bladder is going to sit right on top of the prostate. So this is a really important reproductive gland. It's about the size of a walnut. And so the bladder sits right on top of the prostate gland. It's going to sit right behind the pubic symphysis and right in front of the male rectum. Between the male rectum and the back side of the bladder is another really important pair of reproductive glands, and those are called the seminal vesicles. So I have here just in anatomy, it's anterior to the rectum, superior to the prostate, and posterior to the pubic symphysis. And it's all going to be retroperitoneal. We look at the female, same thing. It's going to be retroperitoneal. So here's that parietal peritoneum coming down and going over it. In females, when we look at the female reproductive structures, we now have a much larger female reproductive structure in the pelvic floor, which is the uterus. The uterus is actually going to sit between essentially the back edge of the bladder, and it kind of leans over the top of the bladder and in front of the rectum. So when we're looking here, I have my pubic symphysis. The bladder is going to sit just behind the pubic symphysis. It's going to sit in front of the rectum and vagina. So this is the vagina here, or vaginal canal. So I have the rectum, the vagina that the bladder is going to sit in front of, and then the uterus is kind of sitting under the back and kind of leaning over the top of the bladder. All right, so I have here anterior to the vagina, it's going to be inferior, most of it, to the uterus, and it's going to be posterior to the pubic symphysis. All right, so collection-wise, the bladder functions to expand, and it has these nice big folds in it called rugae, very, very similar to the interior of the stomach. What we're looking at here, this is kind of a cool view, is you're looking at the bladder as if you cut the front off of it. So it's actually kind of pointed in the front. And so we're going to cut the front off, or the pointy part of it off. And we're looking at, this is the back edge of the inside of the bladder. This is the floor of the bladder, and actually this opening here and this opening here, those are the ureteral openings, and this is the urethra right here. So this will be the inner urethral sphincter, which can contract. If it were a male, we would see a prostate sitting right around the urethra at this location. This smooth section is called the trigone. And again, we're going to see that we have these nice ridges here called rugae. That's going to allow us to expand without tearing. We're also going to see transitional epithelial cells as well. All right, so average volume is usually about half a liter to a liter. A liter is actually pretty optimistic, but you can actually hold a liter if you can stand the stretch receptors. All right, so I mentioned the rugae are these macroscopic foldings that allow distension and stretch, just like the stomach. The trigone is going to be this smooth area devoid of rugae. The idea is when the muscle of the bladder contracts, it kind of stands the bladder upright, so it kind of tips the back end forward. And this functions like a funnel, and it helps to make sure that we drain the majority of urine. We don't want to leave a whole lot of residue, because if you have residue left of urine, it can start to evaporate some of the water, and you can get left with little stones. So we want to make sure we get really effective voiding. So the trigone is this smooth triangular region at the base of the exit of the bladder, and it's marked by the two ureteral openings and the urethra. All right, So this is looking a little bit more depth into the pelvis. So these are the pelvic bones. So this would be the iliac bones on either side. And then you have basically this recess down between the two pelvic bones, which is filled with adipose and a venous plexus, those blood vessels. We see basically this is the back end of the bladder. These are the two ureteral openings. You can see the trigone right here and the urethra. This is male because I see the nice prostate sitting right below the bladder, and this would be the beginning of the male urethra. So this would be what we call the prostatic urethra that then enters into the male penis, and that would be called the penile urethra. All right, so musculature, again, I mentioned that there's 3 layers of muscle. We call these collectively the detrusor muscles. The detrusor muscles allow when your bladder is contracting to get you a really good compression and elevation. So the bladder almost stands upright, so make sure that we void really well. The process of urinating is called micturition, which you can see right here, this term micturition. It's a fancy name, just like deglutition is a fancy name for swallowing. So micturition is urinating. So here you can see as the bladder starts filling, and it fills from the bottom up, we're going to see that we get some stretch receptors that will then provide a sensation about how full it is. So we get this sensation like, oh my gosh, I need to pee or void my urine. Notice that this happens when the bladder's about halfway full. So many of us know that we can say, you know, now's not a good time. You can hold your urine. That sensation goes away until the bladder fills further and you stretch again the receptors. And then we get another feeling of, I need to void my urine. And then you have this opportunity to then use those detrusor muscles. Now you can void even a half-filled bladder, so you don't have to be full before you void. All right, and then last but not least, we have the urethra. And I mentioned in males and females, it's a slightly different pathway out. Both males and females only have one urethra, but in males, we're going to couple it with the reproductive structures. In females, it's solely and entirely used for urine transport. So in females, it's about 1.5 inches to 2 inches long. I mentioned that's another reason why females tend to get urinary tract infections more often, because the urethra, which is just a singular tube here, we have our inner urethral sphincter, and then we have our external urethral sphincter. The inner is smooth muscle, the external is skeletal muscle. So we'll be able to control consciously our external urethral sphincter, and the internal urethral sphincter is going to be determined to contract and relax by smooth muscle or autonomic stretch receptors. All right, males, you have a much longer urethra. So again, we're going to start up here. We have the prostatic urethra and then the penile urethra. And so depending on the individual, maybe between 8 and 10 inches long. It serves for both reproductive substances. So semen will travel this distance as well as will urine. So there's a inner urethral sphincter way up here and an external urethral sphincter as we enter into the male penile urethra. So again, males tend not to get urinary tract infections quite as often, simply because the distance that microbes would have to travel before they can finally enter into the bladder, where we might find urine that has things that the bacteria like to inhabit. All right, so we're going to travel through and past our reproductive structures and then through the male reproductive structure of the penis as we exit. Okay, and I just mentioned micturition is the fancy name for urination, and we're going to utilize primarily smooth muscle in the bladder, but we'll have some skeletal muscle at the external urethral sphincter to make sure that we can avoid at an appropriate time. All right, so again, the inner urethral sphincter is going to be smooth muscle. It's located at the exit of the bladder. And the external urethral sphincter is going to be skeletal muscle. So this is the one we're going to consciously be able to control. And depending on if you're male or female, if you're female, this is going to be right at the exit of the urethra. If you're male, it's going to be right at the entrance into the penis. All right, and so these are just figures of that. So this again is the external urethral sphincter right here at the entrance into the penis. And in females, the external urethral sphincter is way down here, right at the exit from the body. Okay, and this is just another picture of the same thing you guys seen before. So done. Hopefully I'll have a chance to go video what we have in our lab. I'm gonna spend a few hours down there videoing urinary and also some reproductive structure.